Good morning, everyone. We are Group 6 presenting the topic, the structure of chromosomes. First, I'd like to introduce to you our members. My name is Janel Reyes, the leader, Kelvin Idasora, the assistant leader, along with Frederick Karamat and Dancel Cambao as the reporters for today. In today's topic, we will be discussing chromosomes, homologous chromosomes, and the human chromosomes, along with their functions and examples. But before we begin, we would like to show you a video.
Stated Clearly presents What is a chromosome? In previous animations, we learned that DNA is a spiraling chain-like molecule made up of 4 different types of nucleotides. Scientists have labeled them A, C, T, and G. We also learned that a gene is a long stretch of DNA, a specific sequence of A's, C's, T's, and G's that code for something. What does a gene code for? Usually a protein or a group of proteins, though there are a few exceptions. In multicelled creatures like ourselves, proteins, which are coded for by genes, interact with other proteins and molecules to make up living cells. Cells make up tissues, tissues make up organs, and organs ultimately make up entire living creatures. Individuals have different traits, in this case differences in beak shape, largely because of differences or mutations in their genetic code. These differences can either cause changes in a specific protein's shape, and therefore its function, or mutations can change when or how much of a particular protein will be produced. Now that you clearly understand these 3 terms, and by the way, feel free to pause the video to study them if need be, it is now time to talk about chromosomes. A chromosome is an entire chain of DNA along with a group of stabilizing proteins. You may have seen images showing DNA trapped inside a chromosome, sort of like this. This is not how chromosomes hold their DNA. If you were to unravel a chromosome, you would see that it's made of a thread, sort of like a spool of yarn. Zoom in again and you'll see that that fiber is a collection of protein orbs called histones, which are wrapped with a string-like structure. Zoom in on that string and you'll find that this is the chain of DNA. It is extremely long, millions of nucleotides long, and often contains hundreds of genes along its length. In this image, we're looking at a chromosome in its condensed, yarn-like form, but chromosomes are only packaged tightly like this when a cell is reproducing. This structure helps with durability as the cell is splitting in 2. For most of a cell's life, however, chromosomes exist in a loose, noodle-like structure along with other chromosomes safe inside the nucleus or centerpiece of a cell. Here is a photograph of an entire set of human chromosomes taken from a cell right as it was getting ready to reproduce. Your genome, the entire collection of genes that help make you who you are written across 23 pairs of chromosomes. That's 46 altogether. With very few exceptions, each cell in your body contains its own full copy of your chromosomes, your entire genetic code. Eyeball cells only use the eyeball cell genes, the rest are turned off. Liver cells only use liver cell genes, the rest are turned off, and so on for each cell type. One member of each pair of your chromosomes came from your mother, the other came from your father. If you were to examine the 2 individuals of a pair, you'd find that for the most part, both contain the exact same genes in the exact same locations. If you were to zoom into the actual genetic code, however, you would find slight sequence variations between them. These are the results of mutations. Most of these mutations happened long ago and have been passed down from parent to child for many generations. Others, however, are completely unique to you. These mutations occurred either in the sperm or egg, which fused to make you, or they occurred early on in your development, making it into all or many of your cells as you matured. These unique mutations represent brand new genetic information, sequences of DNA that may have never been seen before in the history of the cosmos. So, to sum things up, what exactly is a chromosome? A chromosome is an entire chain of DNA along with a group of stabilizing proteins. Humans typically have 23 pairs of chromosomes in each of their cells. That's 46 altogether. In your body, one member of each pair came from your father, the other from your mother. For most of a cell's life, chromosomes exist in a loose, noodle-like structure. Chromosomes are copied and then condensed like spools of yarn for durability when a cell prepares to reproduce by splitting in 2. I'm John Perry, and that is a chromosome, stated clearly.
Chromosomes are carriers of genes, the unit determiners of hereditary characteristics, tightly packed in the nucleus of eukaryotic cells are the chromosomes. Each chromosome is composed of many structural proteins called histones, which are shaped like a ball and is the genetic material of DNA called around it. Chromosomes are important molecules because they contain DNA and the genetic instruction for the directions of all cell activity. They also carry genes that determine individual traits that can be inherited through reproduction. The DNA molecule is a long coiled structure that carries the genetic instructions on how living things would grow and develop. Portions of DNA in specific patterns are responsible for the physical and heritable characteristics of organisms. These are called genes. The term chromosome came from the Greek word chroma, that means color, and soma, that means body. This is because chromosomes can be stained or colored by specific dyes. DNA is the chemical name for the molecule that carries genetic instruction in all living things. The DNA molecule consists of two strands that wind around one another to form a shape known as a double helix. Each strand has a backbone made of alternating sugar and phosphate groups. Attached to each sugar is one of four bases: adenine, cytosine, guanine, and thymine. Prokaryotic DNA is found freely in the cytoplasm within a region called the nucleus. It is not bound with proteins and therefore it doesn't form chromatin. Genomes are compact and they contain little repetitive DNA and no introns. It contains extra chromosomal plasmids and it is circular in shape. Eukaryotes, on the other hand, is contained within a nucleus. It is bound to proteins called histones. Genomes contain large amounts of non-coding and repetitive DNA, including introns. They are composed of chromatins, each consists of two complementary strands of DNA coiled tightly around histones, and they are linear in shape. One sister chromatid and its parts. When a single chromosome has been replicated in copies, each copy is called a sister chromatid. A special part of the chromosome called the centromere holds the sister chromatid together. When you see an X representing genetic material in an illustration, you are seeing two sister chromatids hold together by the centromere, kind of like if you wanted to keep two sticks together. You might use a rubber band around the center of both. A centromere is like a built-in rubber band for two sister chromosomes. The double-stranded chromosomes called sister chromatids appear as a highly coiled X-shaped structure, joined or attached together at a certain point by a centromere. The location of the centromere on each sister chromatids determine its characteristics, shape, and is used in describing the location of specific genes in a chromosome strand. The centromere is the pinch area in the pair of chromatids that divide the chromatids into sections called arms. The short arm is called the P arm, P for petite, while the long arm is called the Q arm, the next letter of the alphabet after P. Chromosomes are classified according to the location of the centromere along the entire length of the chromosome. Four different types of location have been identified. Telocentric chromosomes have the centromere at the very end of the chromosome. Humans do not possess telocentric chromosomes, but they are found in other species, such as mice. Acrocentric chromosomes have a centromere which is severely offset from the center, leading to one very long and one very short section. Submetacentric chromosomes have the centromere slightly offset from the center, leading to a slight asymmetry in the length of the two sections. Metacentric chromosomes have the centromere in the center, such that both sections are of equal length.
Homologous chromosomes. A pair of homologous chromosomes contains chromosomes of similar length, gene position, and centromere location. Homologous chromosomes should not be mistaken as an exact identical pair as that of sister chromatids. Sister chromatids are products of DNA replication so that each member of a pair is an exact duplicate of the other. Each member of the pair of homologous chromosomes is not the exact copy of the other member. because the pair of homologous chromosomes does not originate from the same organism. They consist of one maternal chromosome and one paternal chromosome that pair up with each other in a cell during meiosis. They are, they are identical only in terms of their structure and the same order of genes in their chromosomal arms. What are maternal chromosomes? Maternal chromosomes are the chromosomes in the nucleus with a maternal origin. This means that these chromosomes come through the female gamete during fertilization. In humans, the set of maternal chromosomes contain 22 autosomal chromosomes in a single sex chromosome. Here, the sex chromosome is always an X chromosome. What are paternal chromosomes? Paternal chromosomes are the chromosomes in the nucleus with paternal origin. It means these chromosomes come through male gametes during fertilization, same as the maternal chromosomes. The set of paternal chromosomes in humans contain 22 autosomal chromosomes. Here, each paternal chromosome has a homologous chromosome in maternal chromosome set. All living things have a characteristic number of chromosomes in their body cells, also called somatic cells. Take note that the number of chromosomes is crucial to the survival of the organism. Any increase or decrease in the number of chromosomes of any species of organisms can cause mutation or instability that can lead to the death of organisms. Here, we will see in the table provided the number of chromosomes of some organisms. Haploid cells. Haploid cells containing only one set of chromosomes. Gametes or sex cells are the most common type of haploid cells. They are produced by meiosis and are genetically diverse. When the haploid cells from the male and female fuse together during fertilization, it forms a diploid cell. Diploid cells. These cells have two sets of chromosomes. It is formed by the fusion of two haploid cells. Most mammals are diploid. They have two homologous copies of each chromosome in the cell. They are produced by mitosis. The skin, blood, muscle cells, also known as somatic cells in humans, are diploid cells. The main difference between haploid and diploid cells is how they reproduce. Haploid cells are formed through meiosis, and diploid cells undergo mitosis. Human and most other mammals are diploid organisms. The major differences are summarized below. You can use the chart for future reference and to easily understand the difference between haploid and diploid cells. A change in the number of chromosomes can cause problems with growth, development, and function with the body system. These changes can occur during the formation of reproductive cells, eggs, and sperm, in early fetal development, or in any cell after birth. A gain or loss in the number of chromosomes from the normal 46 is called aneuploidy. A common form of aneuploidy is trisomy, or the presence of an extra chromosome in cells. People with trisomy have three copies of particular chromosome in cells instead of the normal two copies. Down syndrome, also known as trisomy 21, is an example of condition caused by trisomy. People with Down syndrome typically have three copies of chromosomes 21 in each cell for a total of 47 chromosomes per cell. Monosomy, or the loss of one chromosome in cells, is another kind of aneuploidy. People with monosomy have one copy of particular chromosome in cells instead of the normal two copies. Turner syndrome, also known as monosomy X, is a condition caused by monosomy. Women with Turner syndrome usually have only one copy of the X chromosome in every cell for a total of 45 chromosomes per cell.
The human chromosomes in human Each cell normally contains 23 pairs of chromosomes for a total of 46. 22 of these pair-called autosomes look the same, both males and female. The 23rd pair, the sex chromosomes, differ between males and females. Females have two copies of X chromosomes, while males have one X and one Y chromosomes. Characteristically, the X chromosomes is 3 times the size of the Y chromosomes and carries 100 times more genes than the Y chromosomes. Karyotypes are prepared using standardized staining procedure that reveal characteristic structural feature for each chromosomes. The 22 autosomes are numbered by size. The other two chromosomes, X and Y, are the sex chromosomes. This picture of the human chromosomes lined up in a pair is called karyotype. The study of chromosomes is called cytogenetic. The chromosomes are studied and analyzed by specially trained cytogenetic technologies. It could be observed in this karyotype. Each chromosome is double-stranded. The recent and actual photograph was taken when the cell is in the metaphase stage, where in the metaphase stage, during the process of cell division, usually individual chromosomes cannot be observed in the cell nucleus. However, during metaphase of mitosis or meiosis, the chromosomes condense and become distinguishable as they align in the center of the dividing cell. Metaphase chromosomes are used during the karyotyping procedure that is used to look for a chromosomal abnormalities.
Thank you for listening. Again, we are the Group 6 presenting the structure of chromosomes.